total RBC count. Viva questions. Question 1. How will you differentiate a RBC pipette from a WBC pipette? There are 3 reasons. First, color of the bead. In RBC pipette, the color of the bead is red. Second, size of the bulb. The size of the bulb of the WBC pipette is smaller than the RBC pipette. So RBC pipette bulb size is larger. And third, the readings. The readings on the RBC pipette are 3, 0.5, 1 and 101. So these are the 3 reasons how you can differentiate an RBC pipette from a WBC pipette. Why is blood diluted 200 times for RBC count? The answer is simple, because RBC count is very high. It is in millions. What is the function of a bead in the bulb of an RBC pipette? It has 3 uses. First, it helps to identify the pipette, whether it is an RBC pipette or a WBC pipette. Second, it helps in mixing the contents thoroughly. And third, it tells us whether the pipette is dry or not. Why is it important to discard the first few drops from the pipette before charging? The first few drops are having HAMS fluid in the stem of the pipette. That is why the first few drops are discarded before charging the chamber. What is the function of a cover slip? It has 2 functions. The first function is to control the volume of fluid. Second function, it helps in the uniform distribution of cells by preventing the flow of fluid into the gutters. Why should a broken cover slip not be used? A broken cover slip when placed is placed asymmetrically, and because of that it causes uneven distribution of cells and the count is affected. What are the alternatives to HAMS fluid? The first one is DACI solution. DACIE, DACI solution. Very simple to make, can be stored for a long time. The second alternative is normal saline. It contains 0.9% sodium chloride. It is used when HAMS fluid and DACI solution is not available. There is a problem here. When you are using normal saline, the count should be done within 1 hour of filling the pipette, because after that, rouleau formation is going to occur. Also, for this special purpose, a stock solution of normal saline cannot be kept specially. Why small excess of blood in the pipette should not be removed by a cotton? Small excess of pipette in the blood should not be removed by cotton, because cotton absorbs only the fluid portion, leaving behind the cellular portion. So the RBC count comes high. How should we remove the small excess of blood from the pipette? It should be lightly tapped on the fingers or the palms. How can RBCs and dust particles be differentiated under a microscope? The answer is very simple. RBCs are rounded discs, uniform size and shape, light pink in color with a lighter color at the center. Dust particles are irregular in size and shape. First, they disappear with change of focus, and second, if they are on the eyepiece, they will rotate with the eyepiece. So this is how you differentiate from red blood cells and dust particles. What are other uses of an RBC pipette other than counting RBC count? There are 3 other uses. First, to count sperms. Second, to count platelets. And third, to count WBCs